Well, hey, everybody, this is Robert, and welcome to this show where I'm going to take a look at a little introduction to clinical parasitology, some definitions, and the like. And it won't be all inclusive, but it'll cover a lot of different things for somebody dabbling first with parasitology. So let's go ahead and start out with some definitions, right? Parasitology is the science that deals with parasites. A parasite is an organism living temporarily or permanently in or on another organism or the host, from which it is physically or physiologically dependent upon another. And parasites can be either unicellular, like an amoeba. It can be like a worm, like, say, Ascaris lumbricoides. Or it can be an arthropod. Some of the features of parasites: they are smaller than their host. They outnumber their host. They have a shorter lifespan than their host, typically. And they have a great reproductive potential, much more than the host. Thousands upon thousands of eggs, for example, can be produced by some of these parasites. There's different associations between the different organisms and some definitions. So symbiosis is essentially the term for the association between two different organisms. How do they live together? And those are broken down into mutualism, where there's benefit derived by both. Commensalism is when the parasite is benefited from the host, while the other one, the host itself, is neither benefited nor harmed. And parasitism, of course, is when one organism lives at the expense of the other. Parasites can be classified in a number of ways. They can, by their habitat, inside the body, outside the body, right? Ectoparasites versus endoparasites. According to their dependence on the host, some are obligate. They have to have a host to survive, and some are temporary. And of course, according to the pathogenicity, some are pathogenic, as we already spoke, the parasites. Some are commensal, some are opportunistic. There's different types of host. The definitive host, this is where the host harbors the adult stage of the parasite or the sexually mature stages of the parasite. An example of this I have listed here is humans, or man, is the definitive host of the beef tapeworm, Taenia saginata. An intermediate host is the host that's harboring the sexually immature or the larval stage of a parasite. An example of this would be with the same parasite, Taenia saginata, the cow would be the intermediate host. A reservoir host, this is an animal that harbors the parasite and acts as a source of infections to humans. And then there's the accidental or the incidental host. Infection of a host other than the normal host species. A parasite may or may not continue full development in the host. An example of this is the different schistosomes that can cause swimmer's itch in a person. Some more definitions. The vector. There's biological vector, where part of the parasite's life cycle occurs. The Anopheles mosquito is a prime example, where part of the malaria life cycle occurs. And then there's mechanical vectors. And these are vectors that are not essential in the life cycle. These are passive carriers. The diagnostic stage, this is a developmental stage of a pathogenic organism that can be detected in stool, blood, urine, etc., etc. An example of this would be the eggs of, say, Taenia saginata, for example. The cyst stage or the trophozoite stage of Giardia lamblia. Now, the infective stage is the stage of the parasite which is capable of entering the host and continuing development within the host. An infective stage for, say, example, Strongyloides stercoralis would be the faleriform larva. Infestation, this is an establishment of arthropods upon or within a host. Zoonosis, this is a disease of animals, and these diseases that are transmissible to man or humans. Autoinfection, an infection an individual acts as the source of the hyperinfection to him or herself. Again, strongyloides is a good example. As the egg stage of the strongyloides starts leaving the intestinal rectal area, sometimes this hatches ahead of time and the larva comes out and it's able to penetrate the skin and auto-infect the person. And hyperinfection is strongyloides, again, another good example. This is when the parasite is reinfected by the individual that is harboring the parasite is reinfected by the same parasite. But in this case, when it's hyperinfection, we're thinking about somebody who's immunocompromised in some way, and somebody, say, who is on organ transplant medications or whatever, and they had strongyloides. The strongyloides would keep on reinfecting them until you get to a point where you could have a life-threatening hyperinfection of this parasite. Sources of infection: contaminated soil. That's a common one for the soil transmissible helminths, Ascaris, Trichuris, hookworm, strongyloides, and others. Contaminated water, amoebic cyst, blood flukes like the schistosomes, guinea worm, fasciola, insufficiently cooked meat, which contains infective stage of the parasite. A couple of good examples here are trichinella, undercooked pork, Taenia solium, undercooked pork, Taenia saginata, undercooked beef. Blood-sucking arthropods, the Anopheles mosquito for malaria, the tsetse fly for sleeping sickness. Dogs or animals are an example, and dogs is one example of an animal where something like Toxocara could be transmitted via your pet. Other humans, pinworms is a good example. Sexual intercourse, Trichomonas vaginalis, and autoinfection, which we've already talked about. Two primary modes of transmission. One is the direct mode. A horizontal transmission would be, in most cases, fecal-oral, and the vertical transmission is mother to child. Mother to child would be a good toxoplasma would be a good example of vertical transmission. And then, of course, indirect mode, and that is through the biological vectors, the mosquitoes, the tsetse flies, the kissing bugs, etc., etc. Route of transmission. There's a number of ways that people can pick up parasites, and ingestion's a very common one. Skin penetration, hookworm, strongyloides, the schistosomes. Insect bite, we've talked about that quite a bit already. Sexual contact in the case of Trichomonas vaginalis. Transplacental, like with Toxoplasma gondii. Inhalation, Pneumocystis carinii. And others like transmammary, we see that sometimes with strongyloides. Even kissing, there's certain parasites that can be transmitted via kissing, like Trichomonas tenax or Entamoeba gingivalis. Effects of parasites on their hosts. A lot of different pathology can go on here. The depletion of nutritive elements of the host. Hookworm is a great example. The hookworm attaches to the intestine and drinks blood. And these people can sometimes have iron deficiency anemia. Obstruction, Ascaris lumbricoides. You get enough of these worms that form a bolus in the intestine, you can have intestinal obstruction. Bleeding, great example of this would be Schistosoma hematobium, the bladder fluke, and you can see bleeding in the urine. Destruction of tissues. Entamoeba histolytica can chew through tissues. Naegleria fowleri can destroy brain tissue. Release of toxic substances. The example I put here is the rupture of Echinococcus granulosus cyst, resulting in anaphylactic shock. Secondary infections are always a possibility. Predisposition of malignancy. Again, go back to Schistosoma hematobium. There may be some predisposition of cancer, bladder cancer, with Schistosoma hematobium. Clonorchis sinensis may be a predisposition for liver cancer. So those are some examples. There's host susceptibility factors, and not all parasitic infections cause disease of clinical significance. Both the host and the parasitic factors are involved. Some of the host factors include genetics, age, sex. How immune are you? Do you have a good immunity? Malnutrition. A lot of these diseases cause very severe disease in places where children are severely malnourished. Of course, the intensity and the frequency of infections, the presence of a coexisting disease. HIV is a very good example. Most people that are immunocompetent are not going to have an issue with a parasite like pneumocystis. Lifestyle and occupation, another good example. Occupation, people that work around different meat and stuff like that would be an example. Then there's the parasite factors, the strain of the parasite, the parasite load, how many parasites did you get infected with at one time, and the sites occupied in the body. Obviously, a parasite in the central nervous system is going to be very severe as opposed to other parts of the body. Types of specimens used for parasite exam, and they can be just about anything, but some of the more common ones would be stool, blood, urine, sputum for example, lung flukes would be a good example, Paragonimus. CSF, some of our free-living amoeba we can find in the CSF. Skin, bone marrow, lymph nodes, liver aspirate, muscle biopsy. Trichinella spiralis can be found in a muscle biopsy. Duodenal aspirates, Giardia, and other parasites can be found in that. From the perianal folds, pinworms, for example. Classification. Now there's 3 primary phyla of human parasites, and that's the protozoa, the platyhelminths, which include the tapeworms and the flukes, and the nemohelminths, which are the roundworms. Now the protozoa are broken down into generally 4 categories, and a lot of it's based on their method of motion. So amoebas, which get along mostly by pseudopods, flagellates like Giardia, flagella, and ciliates have little cilia all around the edge of their shell like Balantidium coli. Sporozoa are typically non-motile, but they get around by, a lot of times, by a vector. Say malaria gets around by the mosquito. Other ones do not have a biological vector, like, say, Cryptosporidium. And then there's the helminths, and we kind of touched on this already, but there's 3 major categories: the nematodes or roundworms, the cestodes or tapeworms, and the trematodes or flukes. I want to briefly go over some laboratory procedures. I mean, this is really very basic stuff, but this is a good primer for laboratory procedures. Stool should be collected in a dry, clean, leak-proof container. Make sure it's not tainted with urine or water or soil, anything else that could cause things that could interfere with the stool exam or even other parasites. Fresh stool should be examined and processed and preserved immediately. If you have trophozoites in a fresh stool, you should look at that pretty quickly, like within 30 minutes, while the trophozoa is still viable. If you can't examine fresh stool in a timely basis, you should preserve the specimen. And there's various preservatives that are available out there. Some of the most common ones are 10% formalin, polyvinyl alcohol, or PVA. There's also SAF and several others out there on the market. And on this little diagram here near my head, you can see where the more formed a stool is, the less likely you are to see a troph, while the more watery the stool is, the more likely you can find a troph stage of a protozoa. Now, if kits are not available, the specimen should be divided and stored in 2 different preservatives. 10% formalin, and PVA. You can add 1 volume of the stool to 3 volumes of the preservative. And these are just some advantages and disadvantages of the 2 preservatives we're focusing on in this video. And formalin, really an all-purpose fixative, long shelf life, good preservation and morphology of helminth eggs, larvae, protozoan cyst, coccidia, suitable for acid-fast stains and others. Disadvantages is they're not really good for permanent mounts like the trichrome stain, and they're not very good for the preservation of morphology of the protozoan trophozoites. The PVA, good preservation of morphology for protozoan trophozoites. It's good for permanent mounts like the trichrome stain or the iron hematoxylin stain. Some of their disadvantages, inadequate preservation of morphology of helminth eggs, etc. It does contain a mercury compound, which makes it hard to dispose of and expensive to dispose. And it can't really be used for various stains like acid-fast and others. Examination of the stool sample, protozoan trophozoites, cyst, oocyst, and helminth eggs and larvae may be seen and identified using a wet mount identification technique. To prepare the wet mount, obtain a microscope slide and the stool specimen. Take a small amount of the stool, place it on the slide. If the stool specimen is still somewhat solid, add a little bit of saline and mix it up. Ideally, 2 smears can be prepared on one slide, one with iodine, one without, and you can actually seal it using petroleum jelly or paraffin. And you can see here on the right-hand side above my head that it shouldn't be so thick that you can't read newspaper print through it. The direct examination of a saline prep, scan on low, identify on high power. Iodine, this is why you want to use a non-iodine prep and an iodine prep, because the iodine could destroy the trophozoite stage. Iodine is very useful though in staining cytoplasm, yellow-brown. Parasites like Iotamoeba bushlii, the glycogen vacuole will stain a very deep brown, it's a very, very good technique for identifying that parasite. It's very unique and you really can't miss it. And you always want to use a micrometer on your microscope so you can measure the size of the parasite. And here's a couple examples. On this side, it looks like an entamoeba, and you can see how the iodine is accentuating the peripheral chromatin here on the nuclei. And on the one on the left, you can see how the glycogen vacuole, this Iotamoeba, is staining a darker brown color. Permanent stain mounts. This is mostly for protozoa, and use fresh stool or PVA. Trichrome stain and iron hematoxylin are 2 of the most common ones. On the right-hand side, you can see a trichrome stain. The nuclear chromatin stains red-purple. Chromatoid bodies, if they're present, they'll be kind of dark red. The cytoplasm stains kind of a greenish. If there's charcoal-laden crystals in the stool, they'll be a very bright red. And if there's a glycogen vacuole or in the parasite, they will remain clear. They won't pick up the stain. Iron hematoxylin, a darker type of stain. You see a blue-gray cytoplasm, dark blue or black nuclei and chromatoid bodies. If there's charcoal-laden crystals, again, blue-black. And in the case of this parasite here, another Iotamoeba, the glycogen does not pick up the stain, so it should be clear. Concentration of stools, there's a couple different methods, but the concentration methods are to separate the parasites from the fecal debris, which allows you to have a better chance of identifying the parasites, even when they're in small numbers. And there's two techniques. One is the flotation techniques and one is the sedimentation techniques. Flotation, zinc sulfate's a very common one, for example. You use a zinc solution that has a higher specific gravity than the organisms. And so the organisms actually rise to the top while all the debris sinks to the bottom. And you put a little cover slip on the slide and the parasites will be there. This is a good technique for a very clean looking parasite slide. There are some disadvantages, and not a lot of clinical laboratories use this method because this technique does do some damage to the wall of cysts and eggs, which can affect you trying to identify them. If the egg is a percolated, that'll drop to the bottom, so you'll miss that. And for those larger parasites that have a higher specific gravity than the zinc, for example, that will also drop to the bottom. Sedimentation. This uses solutions of a lower specific gravity than the parasitic organisms, so the concentrating of the parasites remain in the sediment. It's recommended for general diagnostic laboratories because they are easier to perform and less prone to technical error. A common technique is the formalin ethyl acetate, and you can see that right here on the right. All the debris right here, then you got your formalin ethyl acetate is at the top, and here, after you decant all this, this is what you're going to be working with, is this sediment down at the bottom. Blood specimens, I mean, we have a lot of different blood parasites, and this is how you can identify them. They should be collected prior to treatment. Blood smears should be obtained and examined without delay, since the parasitemia may fluctuate. Multiple smears might need to be needed. These can be taken at 8 to 12 hour intervals for 2 to 3 days. Finger stick is preferred for, say, malaria. A lot of the different anticoagulants will interfere with the parasite morphology and the staining, so that can present a problem. If you have to use venipuncture, try to make the slides at the time of phlebotomy. And if you have to collect a sample, which often happens, EDTA is the preferred anticoagulant. There's going to be thick and thin smears made for many of the parasites, the blood parasites. Thick smears will have multiple layers of blood, as you can see on the slide. It's a big, thick globule. They'll be lysed, and then you have all these layers of lysed RBCs, which allows you a better chance for detecting the parasite, an increased sensitivity. But it's not typically ideal for identifying the parasite because the morphology is not going to be necessarily typical. And in the thin smear, which was on the other side of that slide, it's kind of like making a slide for a white cell differential or something like that. And this is an even spread layer where it's feathered at the end, so the thickness continually decreases. So it should be at some point a monolayer of RBCs not touching one another. And this is the slide you would want to use to speciate your parasite. If you have a bunch of Plasmodium, for example, on the thick smear, you're going to want to speciate it on the thin smear to determine, is it falciparum, is it vivax, etc., etc. And you stain these with Giemsa or Wright stains. Now, microfilaria are kind of a unique situation because of their periodicity, and that's all dependent on the species of parasite we're dealing with. So essentially, the parasite comes out at different times of the day or night, and collecting blood at that specific time allows you a better chance of demonstrating the microfilaria. And I wrote a couple examples here. So Loa loa, the African eyeworm, is best found during the midday, 10 a.m. to 2 p.m. While the parasites that will cause filariasis, Brugia malayi, Wucheria bancroftii, it's best to collect at night after 8 p.m. The modified acid-fast stain, which a lot of you know from bacteriology and for identifying acid-fast bacilli like tuberculosis, is also used for some parasite techniques. And it's really useful for identifying the oocyst of some of the coccidian species. For example, Cryptosporidium, Cyclospora, Cystoisospora, which may be hard to see in a routine trichrome stain, and because of their size, may be a little bit more difficult for inexperienced techs on say a wet mount or a sedimentation concentrate. And that's it. That's my little intro to human parasitology. Got some definitions, some procedures. So hopefully you learned a little bit out of this, and I appreciate you watching. Please subscribe to the channel. Share this with your friends. If you're in a medical technology program, share it with your fellow students. And I'll see you next time on Outbreak News TV.